And I think I will take over. Thank you both. Let me share my screen here. PowerPoint. I think that should work. Hopefully, yes. So it's my very great pleasure to introduce Ann Andrews, who's a professor of psychiatry and behavioral sciences and chemistry and biochemistry at UCLA, where she held the Richard Metzner Endowed Chair in Clinical Neuropharmacology. She's a member of the Semel Institute for Neuroscience and Human Behavior, the Hato Center for Neuropharmacology, a number of other brain institutes, and the California Nanosystems Institute. As you heard during the panel, she began her career at the National Institute of Mental Health, moved to Penn State, and then to UCLA. She's also president of the International Society for Serotonin Research. She served as an associate editor of ACS Chemical Neuroscience, has won awards from NIH, NARSAD, and the American Parkinson's Disease Association, among many others. She was a pioneer and a champion for applying advances in nanoscience and nanotechnology to neuroscience, including the BRAIN Initiative in the US, which we'll hear about today. I'll add that Professor Andrews is my favorite scientist, so it's a special pleasure to introduce her here. And the floor is yours.
Thank you, Paul, for that really warm introduction. I'm hoping you can see my screen. Yes?
Not yet.
Okay. Let's see. Oops. See. Let's go back. Oh, here. Let's try to escape. Let's see if we can try again. Okay. All right. Let's do this first. Give me one sec.
There we go. Not presentation mode yet, though.
All right. We can fix that. And how's that working? Presentation mode?
Yes. Great.
There we go. All right. Well, it's a real pleasure to be here today. I have to say that I hear Paul get up for these lectures, and he's always so excited by the speakers, by the audience, and it's really a pleasure for me to be here today. It's quite early in the morning here in Los Angeles, so I'll say good morning, but for many of you, it is a good evening, a good Friday evening. The last question that Ed got on how might we use optogenetics to interface with multiplexed neurotransmitter sensing, I hope that I'm going to answer that question for you. So thank you to the person who asked that question. I guess I wanted to start here with this idea that many people think that space is really one of our great final frontiers. And we were really excited this year when our friend and colleague, Professor Andrea Ghez, won the Nobel Prize for her work on imaging in space. She was able to image dark matter in a black hole in the center of our universe, and it's an illustration of how imaging can teach us about our world. However, as you heard in Ed's talk, I really believe that we have another final frontier, and that final frontier is our own human brains. It's that 3 pounds of tissue that sits in our skull. And of all of the systems in the body, the brain is the system that we probably know the least about. We can probe its structure. We can even image some function in humans using functional magnetic resonance imaging. But yet we still really understand very little about how complex behaviors like emotions and thoughts and memories arise from the function of the brain. Now, scientists have been studying the brain for well over 100 years. And as Ed talked about, imaging, just like in space, has been critical to our understanding of the architecture of the brain. And so going all the way back to the early 1900s, some of the first images of the cells in the brain were made by two scientists, Santiago Ramón y Cajal, who was a Spanish scientist, and using a technique that had been developed by Camillo Golgi. And you can see some of their early images on the left. These are Purkinje cells from the cerebellum, and they were imaged by the light microscope. And for this work to determine that the unit of function in the brain was the neuronal cell, these two scientists were awarded the Nobel Prize also. However, these early physiologists, early anatomists, we didn't really even call them neuroscientists back then, they quickly developed an argument amongst themselves having to do with how these neuronal cells communicated with one another. You saw some beautiful images in Ed's talk of our current ability to image and to record electrical transmission. And we now know quite a bit about how neurons transmit information along their processes in the form of an electrochemical signal. However, the early images of neurons in the brain were not resolved enough to tell us whether those neurons actually physically touched one another. And so these early neuroscientists argued as to whether electrical signals could pass directly between neurons or whether there was some type of intermediate signal that was used for interneuronal communication. And this became known as the War of the Soups and the Sparks. The smaller group, the Soups, made the argument that in fact neurons might not touch each other, and there was a possibility that they were releasing some kind of substance into the space between them that was used for chemical communication. And this argument between the Soups and the Sparks was eventually resolved by pivotal experiments that were done by a German scientist named Otto Louis. What Louis did was he dissected hearts from a frog. And in the case of the first heart, he retained the vagus nerve, which is connected to the heart. And when stimulated, the vagus nerve slows down the heart rate. He stimulated the vagus nerve, and then he took the fluid from around the first heart and he pumped it into the bath surrounding a second heart. And what he discovered was that by simply transferring that liquid bath substance, he was able to slow the intrinsic beating of the second heart. And that was evidence that indeed a chemical substance was released by the heart cells in response to this neuronal stimulation, and that substance could diffuse and affect other cells. And Louis was also awarded the Nobel Prize for his discoveries in combination with Sir Henry Dale. Of course, we now know that there is a space between neurons. The first neuron we can refer to is the presynaptic neuron, and the presynaptic neuron communicates to a postsynaptic neuron across space, which we call a synapse. This space is on the order of about 10 to 20 nanometers. And so signaling in the brain has always been a nanoscale endeavor. And we now know that presynaptic neurons, when electrically stimulated, release chemical substances into the extracellular space. And these substances not only diffuse across the synaptic gap, but they also diffuse out into the extracellular space to communicate with many other neurons. The first neurotransmitter that was discovered by Louis was this neurotransmitter here called acetylcholine. Today, we know of many different substances that have either been confirmed or are suspected to be chemical messengers in the brain. In fact, people hypothesize that there may be 100 or more different substances that are used for interneuronal chemical communication. And one of the things that's fascinating about these chemical messengers or neurotransmitters is that they come from all different chemical classes of molecules. You can see that some neurotransmitters are amino acids, others are derivatives of amino acids, and I'll focus a little bit in my talk today on this neurotransmitter here called serotonin. Some neurotransmitters are derivatives of steroid molecules, others are free fatty acids. And the largest class of neurotransmitter molecules, which is still largely unexplored, are the neuropeptides. And so if we wanted to investigate how information is encoded in chemical signaling in the brain, we would need to develop methods that would enable us to measure all of these very highly different types of molecules. And this is a truly difficult chemical challenge. In addition, not only do we need to be able to differentiate molecules that are widely different in terms of their chemical class, we also need to be able to differentiate molecules that are very similar in structure. For instance, this neurotransmitter here called dopamine, which many of you may have heard of, and another neurotransmitter called norepinephrine or noradrenaline, which, as you can see, differs from dopamine only by a single hydroxyl group. So we have at the same time this challenge of being able to try to sense many different types of molecules, yet to be able to sense molecules with high selectivity, if we want to indeed think about understanding how all of these different neurotransmitters are working. And in fact, Ed mentioned that one way to think about the brain is to think about the brain like a computer, digital signals on and off, ones and zeros. But I would actually challenge you to think about the brain more like a piece of music. And in fact, all of these different neurotransmitters are operating in the brain at the same time, much like when we listen to a piece of music, there are many different instruments, many different tones that are all being played simultaneously. And so, unlike a computer, the brain is really a many-layered analog system. So as Paul mentioned, I had the great privilege of being involved in developing the Brain Initiative, which was announced by the Obama White House. And we have been working together, many of us now who have joined the initiative, to try to develop neurotechnologies with the goal of being able to record information in the brain at ever larger scales, at ever finer detail, and to try to use these large data sets to be able to understand the emergent properties of how information is encoded in the brain. And for my part, what I tried to bring to this endeavor was the idea that we would need to focus on the chemical signaling, that we would be unable to truly understand how brains function if we only focused on the electrical signal and we ignored this really rich, analog, multi-layered chemistry in the brain. And if we can put that all together, we'll be able to understand the brain in what I've termed its chemical connectome. So in my own group, we focus on the neurotransmitter serotonin. You can see its structure here again. And many of the methods that we use to study serotonin signaling in the brain involve electrochemistry. Serotonin is easily oxidized, and when that happens, it loses 2 electrons, and we can measure that loss of electrons as a change in current. And using Faraday's law, we can then understand how current is reflected in change in concentration of serotonin. The serotonin system is complicated. It's involved, we think, in a number of important neuropsychiatric disorders. And for the research that's carried out in my own laboratory, we focus mainly on trying to understand how serotonin encodes information involved with anxiety and mood. One of the proteins that we have focused on in our research is the protein that you see here in green, and this is the protein called the serotonin transporter. And the function of this protein is to take serotonin back up into presynaptic neurons after it's been released. So neurotransmitters are recycled, and this is biologically efficient. And many of you may be familiar indirectly with the serotonin transporter because this is the main molecular target for the largest class of antidepressant and anti-anxiety medications. These drugs are called the serotonin selective reuptake inhibitors. And they're drugs like Prozac, Paxil, Zoloft, Lexapro, and they're widely used in the treatment of anxiety and depression, as well as a number of other disorders. So I wanted to just spend 1 or 2 minutes showing you a technique that we use in our laboratory called fast microdialysis. This is a mature technology, and we've contributed to the development by vastly improving the temporal resolution of this technique. You can see that we can perform measurements using fast microdialysis in awake behaving animals, as seen in this video. The technique involves implanting a small membrane into the brain, which you see up in the left-hand corner. We perfuse into this membrane artificial cerebrospinal fluid, and then neurotransmitters like serotonin or dopamine can diffuse from the brain extracellular space into this semi-permeable membrane. They're swept out of the membrane, and then they're taken directly into a high-performance liquid chromatography instrument coupled with electrochemical detection. And we can measure changes in serotonin concentrations in very small locations in animals' brains as they're behaving. And we have resolution for these measurements now on the order of about a minute per measurement, which you see here in this graph. We can also deliver stimulus through the microdialysis membrane by infusing artificial cerebrospinal fluid that contains a high concentration of potassium. And that causes neurons to fire. So every time we deliver potassium, you can see that the serotonin concentrations increase. And the longer the potassium stimulation, the larger the increase in extracellular serotonin. Using this type of method, we've been able to investigate what happens when you give an SSRI antidepressant to animals. And as you can see in the blue trace, so this is from an animal that was locally infused with the antidepressant citalopram. What happens is that when we stimulate, we see a large overflow in extracellular serotonin compared to an animal here in black that did not receive this drug. So in this way, we're able to study in real time and in conjunction with behavior how different types of drugs function in terms of their interactions and their ability to impact chemical neurotransmission. This type of methodology is also capable of multiplexing because we can separate different analytes in the HPLC. So recently we've been doing fast microdialysis for both serotonin and dopamine. And we've also been able to couple instead of high potassium stimulation, optogenetic stimulation with this type of method. I won't show those data, but we're working on a manuscript right now reporting on those results. And the development of fast microdialysis was largely done by Dr. Hongyan Yang, who you see pictured here in the lower right. However, there are some downsides to the microdialysis method. The first is that while that membrane is small, it's on the order of about 250 microns in diameter and about 1 to 2 millimeters in length. The size of that type of probe is still rather large compared to a number of different brain structures that we would like to investigate. Secondly, the temporal resolution of microdialysis is likely limited to the minute range because of different limitations having to do with the instrumentation, with diffusion. We're never going to be able to make faster measurements using that kind of method. And so as far back as really about 20 years ago, I began to try to think about how we might make a really large step forward in terms of being able to make measurements of neurochemicals in the brain. I wanted to think about how we might develop a truly next generation type of sensor that would enable us to make fast measurements on the order of seconds or milliseconds, a methodology that would enable us to measure multiple neurotransmitters, and one that would be highly spatially resolved. What you see pictured here is a probe. This is a silicon probe that's micro-manufactured by my colleague Sotiris Masmanidis, who you see pictured in the right. And this type of probe is used for measuring electrophysiology. So each of these pads or measurement sites that you see, these are metal sites. This probe can be fabricated with up to 1,000 different recording sites. And each of these recording sites can be used to measure electrophysiology in the brain. And so in thinking about how we might advance our measurements of neurochemistry, we started to think about how we could take devices like this or other types of micro and nanoscale architectures and impart them with some type of molecular recognition that would enable these sensors now to be able to detect neurochemicals. And so I'm going to talk to you about our efforts in this area. And we have settled in on a design that you see here. Instead of using metal electrodes, we're now going to replace on these silicon microprobes those recording elements with field effect transistors. And of course, field effect transistors, or FETs, are the basic unit of function in all of our electronic devices, our laptops, our cell phones. But these field effect transistors are most commonly used in dry environments for processing electrical signals. And so how might we take these field effect transistors and turn them into something that can be used as a chemical sensor? And there were some hints in the literature that one could couple biological macromolecules with the semiconductor portion of a field effect transistor. And when the biomacromolecule would recognize a target, then a conformation change happens, and that impacts the electric field near the surface of a semiconductor. And we can detect that change in electric field between the source and the drain electrodes as a change in conductance. Now, most of the work that had been done in this area in the early 2000s was using proteins as the molecular recognition element. But I decided very on that there might be a good reason not to focus on proteins, but instead to use nucleic acids for molecular recognition. And other scientists had learned that it was possible to identify rare single-stranded nucleic acids, small sequences on the order of about 30 to 50 nucleotides, that when they adopt their native three-dimensional conformation, they can recognize, they can bind to small molecules. And this had been learned in natural systems through the discovery of something called riboswitches. We can also evolve or identify in the laboratory rare sequences, and those rare sequences are called aptamers. It had been shown, for instance, that an aptamer was identified that could differentiate this small molecule drug called theophylline, which is used for the treatment of asthma, that an aptamer could identify and differentiate theophylline from its closely structured cousin caffeine. So there was precedent in the literature that these small nucleic acids could carry out molecular recognition and that they could do so with high selectivity. And so this adventure in trying to develop these aptamer functionalized field effect transistors has been a really exciting journey, and a lot of it has been carried out in collaboration with Paul. And you can see us on one of our early adventures in Thailand riding elephants. We've learned that aptamers have a number of really distinct advantages when coupled with Fett's for electronic target detection. And one of those advantages is specific for enabling us to take field effect transistors and to make measurements in biological systems. And so when we think about, for instance, the brain extracellular space, this is a high ionic strength fluid. And we can calculate using the equation for the Debye length. The Debye length is the distance above a semiconductor surface over which we can sense a change in electric field. And we can calculate the Debye length in high ionic strength biological fluids. And we find that that length is under 1 nanometer. What that essentially means that if binding to target takes place far away from the semiconductor, for instance, as would happen if we were to use an antibody for molecular recognition, little to no signal change could be detected at the field effect transistor working in vivo in biological fluid, which cannot be diluted. And so by switching molecular recognition to an aptamer, we were able to show that we could carry out these types of measurement. And that's largely because not only are aptamers small, but because they're oligonucleotides, they are densely packed with negative charge. For every nucleotide, we have a single negative charge. And so this high density of negative charge, when target binds, that rearrangement of that highly negatively charged aptamer, some portion of it happens in close proximity to the semiconductor, and we are able to detect that then as a change in transconductance. The first experiments we carried out using aptamer functionalized Fetts were to sense the neurotransmitter dopamine. There was an aptamer that had been identified in the literature, which you see here, for dopamine. We were able to attach that aptamer through a thiol linkage to indium oxide, which we used as our semiconductor. And in work led by a postdoc in the lab at the time, Jay Kim, Jay was able to show that we could sense dopamine down to very low concentrations in this case, down to about 10 picomolar. Now, Ed talked about how important failure is. One of the things that's also really important in scientific investigation is being your own critic of your data and doing a lot of control experiments. And so when Jay showed me these data, these were really unprecedented in terms of the level to which we could sense dopamine. These levels were much lower than anybody had sensed previously using other methods. Not only that, these picomolar concentrations were orders of magnitude below the dissociation constant of this aptamer for dopamine. And so it didn't really seem possible to me that these data could be correct. And I sent poor Jay back to the lab many times with many different control experiments, every one that I could possibly think of. And they all checked out. They all seemed to indicate that, in fact, this was dopamine sensing. And so we went ahead and published this paper in ACS Nano, actually, I think in 2015. One of the things that we were not able to do using this particular dopamine aptamer was to differentiate dopamine from that closely structured neurotransmitter that I showed you earlier called norepinephrine. One of the other things that plays such, I think, a great role in science is serendipity. We've all heard of the phrase that chance favors the prepared mind. And that's really what serendipity was about. So at the time we were doing these experiments, I had the opportunity to travel all the way to Finland, which, as you know, is near the Arctic Circle here above Europe. And it was the fall, it was already cold and dark. And I was visiting the University of Jyväskylä to give a talk at their nanoscience days. And at the end of the day, one of the things that I really miss about in-person talks is that all of the speakers were standing around getting their pictures taken. And I was standing next to a leader in nanoscience from Europe. He was a tall gentleman and he was looking down at me and he said, you know, we tried to use aptamers to do neurotransmitter sensing and we failed. And I thought to myself, well, if he failed, how are we going to succeed? But this gentleman turned to me then and he said, you know, I heard that there is a scientist at Columbia that's really good at identifying aptamers, new aptamers. And I immediately got his name. And the minute I got back to UCLA, I called him. That professor's name was Milan Stojanovic. And indeed, Milan and his right-hand woman, Dr. Cassie Yang, were indeed excellent at identifying aptamers. And he provided me sequences for a dopamine aptamer, a new dopamine aptamer, and a new serotonin aptamer that they'd already identified. We were able to order them, and within weeks, we were making measurements. And indeed, these sequences worked really well for us. You can see here the fluorescence measurements that they had made to characterize these sequences. Already we can see that this new dopamine aptamer is very highly selective. When we coupled these aptamers with the indium oxide thin film field effect transistors, we were able to do really beautiful sensing. Here you can see using the new dopamine aptamer in full strength physiological buffered saline, we can detect dopamine over about 6 orders of magnitude with a detection sensitivity in the low femtomolar range. And for comparison, you can see the data with the new dopamine aptamer in full strength PBS compared to that old dopamine aptamer that I showed you earlier. In this case, we had to dilute the PBS tenfold to even get a signal. And for the new dopamine aptamer, we were now able to sense in undiluted physiological fluid. One of the other advantages of using nucleic acids for molecular recognition is that it's really simple to scramble the primary sequence. And in doing so, you end up with a sensor that's almost identical to the active sensor. But when you scramble the sequence, you can see that we're not able to detect any target at all. So this is a really good way to run controls and to do parallel sensing. We carried out sensing for dopamine in full-strength artificial cerebrospinal fluid. Again, we saw about 6 orders of magnitude of detection with sensitivity to femtomolar ranges. And the same was true using this new serotonin aptamer in full-strength artificial cerebrospinal fluid. We also tested the selectivity of these aptamers on Fett's for closely structurally related targets that we would expect to see in the brain extracellular space. Here you can see that the dopamine aptamer is almost completely selective against serotonin, against that closely structurally related neurotransmitter norepinephrine. We can also differentiate dopamine from its precursor L-DOPA and from its metabolite DOPAC. And the same is true when we tested the serotonin aptamer on field effect transistors. It can differentiate signals versus other neurotransmitters and the precursor and metabolite to serotonin. And this work was led by a graduate student in the lab at the time, Nako Nakatsuka, who you can see completing her first marathon here. We then took serotonin aptamer functionalized Fetts, and we wanted to see how they might perform in brain tissue. And so in this case, you can see the original sensing curve here is here in purple. So we took brain tissue from mice. These mice were genetically engineered so that they didn't synthesize serotonin. And then we were able to add serotonin to the tissue in controlled amounts to carry out the sensing. Then we exposed these sensors after 12 hours, again, to different concentrations of serotonin. And you can see that the sensors perform very well. And so the one thing we did see here was that this curve was now shifted to the right to lower concentrations of serotonin. So we do see some biofouling of these sensors, but it turns out that because they were super sensitive to start, once this curve is shifted in tissue, we actually find ourselves in the range of serotonin that we expect to see in the extracellular space in the brain, which is outlined here in yellow. So it really helps to have a sensor that is too sensitive, if you will, to start. And you can always shift things to a lower sensitivity range. We were able to understand some of the mechanism of action of these sensors by looking at the transfer curves. In this case, we're looking at the voltage sweep between the source electrode and the liquid gate. We then look at how these curves, how these gate voltage sweeps impact source drain current in the presence of increasing concentrations of target. And what we learned was that for some targets, like dopamine or glucose, in the case of a glucose aptamer, as we increase the target concentration, the currents go down. Whereas for other targets, here we see the data for serotonin, or for another target that we investigated, this is a membrane lipid. As we increase the target concentration, the source drain current goes up. And so from these and other data, we were able to come up with this working hypothesis. And that is that some aptamers, when they bind target, a part of the aptamer then changes conformation such that we have more of the aptamer backbone closer to our semiconductor surface. We're using an N-type semiconductor, and so that increase in the density of negative charge restricts the flow of electrons, and that reduces current. On the other hand, some aptamers, when they bind to their targets, they change conformation such that a portion of the highly negatively charged oligonucleotide backbone moves away from the semiconductor surface. And this decreased negative charge density then makes it easier for current to flow. Most recently, we've been looking at oligonucleotide functionalized Fets for the purposes now of detecting hybridization of complementary DNA. So in this case, we're not using oligonucleotides to recognize small molecules in terms of aptamers, but we're using a target oligonucleotide strand functionalized to the semiconductor as a way to detect hybridization of a complementary strand. These data you can see here. When the strand is fully complementary, we can see that the calibrated response of the Fett comes up with time and then it stays high. However, if we add a sequence that's fully non-complementary or mismatched, we can see an initial rise in sequence that then resolves to a negligible change in current. And we've used this approach and we've been able to differentiate target DNA that differs by as little as a single base pair mismatch. In this case, you can see data for a mismatch in the target DNA at the 5th position from the semiconductor functionalization. And we've also investigated how changing the distance of the mismatch from the semiconductor impacts the signal. And we can detect mismatches that are as far as 15 nucleotides from the point of attachment. We've also used this method to look at hybridization between DNA that's attached to the Fett and hybridization with complementary RNA. And just like for DNA hybridization, we can detect fully complementary RNA sequences, and we can differentiate those sequences from other RNA sequences that differ by as little as a single base mismatch. And so we're now looking to the future to try to use this platform as an electronic detection platform to detect specific DNA or RNA from infectious agents, for instance, SARS-CoV-2. We have been developing the electronics to go along with the Fett measurements. So all of the data that I've shown you thus far were collected on an analytical probe station. This kind of measurement device costs about $70,000 US. It's large and bulky and for the most part can only measure from one Fett at a time. But if we think about wanting to measure multiple neurotransmitters, it would mean that we would have to make measurements from multiple Fets at the same time. And so we've been developing instrumentation and electronics to be able to make multi-Fett measurements. Since those electronics currently don't exist, you can see a really early attempt here using something as simple as an Arduino. And we're now in our 3rd generation of printed circuit board. And we are working on trying to measure from 8 Fets simultaneously. And this work is being done in collaboration with Professor Miguel Alcañiz-Filol at the University of Valencia in Spain, and Liv Heidenreich, who's a graduate student. We are also working on fabricating field effect transistors on devices that will be able to be implanted in brain tissue. And so just like those silicon microprobes that I showed you for the electrophysiological experiments, we've been designing and fabricating silicon neuroprobes that now are fabricated to have field effect transistors at their tips. You can see the fabrication scheme here on the left. In this case, only the field effect transistor at the tip is exposed to the brain tissue, and the rest of the shank, which penetrates down into the tissue, is covered with parylene to protect it. And then we can see the source and drain electrodes are wired here. This work has been carried out by a fantastic graduate student in the lab, Chuanzhen Zhao, who you see here on the right. He's getting ready to finish his PhD shortly and start his postdoc. And the work's been done in collaboration with Professor Halman Buket at UCLA and his graduate student, Brenda. Chuanzhen has been able to work out the fabrication for these silicon neuroprobes at the wafer scale. You can see they are quite small. He's fabricated probes that are about 150 microns in diameter. You can see one of those devices here with a 2 Fett design. And he's also fabricated silicon microprobes that are as small as 50 microns in diameter. So quite small and really about the size that we need to go into the brain and make highly spatially resolved measurements. As we reduce the scale of these field effect transistors, they continue to show good electronic properties. Here's an example of an early experiment that we've carried out to do measurements in the brain. These mice are awake. They're trained, just like the mice in Ed's experiments, to be head fixed. So they have 2 stainless steel bars that are surgically implanted to the skull. We train every mice gently by hand so that they're able to be held still by these bars. And then we can drop our neuroprobe into the brain tissue along with a stimulating electrode and a reference electrode to make measurements. And we're in the process of writing up our first recording data using these probes. So I hope that you'll see that in publication very soon. And finally, one of the downsides of using hard material like silicon is that it's not particularly amenable for making long-term measurements. And so if we want to think about measuring neurotransmitters over time to learn something about behavior, for instance, as an animal learns a task, which may take a couple of days or even weeks, we need to start to think about how we might make soft materials that have properties that are more similar to brain tissue itself. And so this is an example of our first attempt at fabricating these thin film indium oxide field effect transistors on a soft polyimide material. You can see that the fabrication can be carried out. The FETs can be delaminated. They're conformal on soft materials. And in this case, this work was done in collaboration with Yang and Professor Tseng. We were able to use unfunctionalized FETs to make measurements that involve changes in pH. We could also functionalize FETs with glucose oxidase and make electrochemical or electronic measurements of glucose. In our most recent work, Chuanzhen has been collaborating with Professor Chongwu Zhou at University of Southern California. They have fabricated thin film field effect transistors on a soft, flexible material called PET. This is a DuPont material. And we know this material because it's used to make a lot of our disposable plastics. You can see that we can now multiplex. We can fabricate a number of FETs in a single device. Here we have the gate electrode and 4 FETs, and then a temperature or a thermistor here to measure body temperature. And these are conformal devices. They can be wrinkled, they can be crumpled, they hold up really well. And just to show you what we can do when we start to multiplex measurements, here you can see at 1 FET that's been functionalized with the aptamer for dopamine. When we add 1 picomolar dopamine, we see that the current at that FET drops, which we would expect now that we know that mechanism of action. However, we don't see a change in the current at a nearby field effect transistor that's been functionalized with an aptamer for serotonin. The same is true when we change the serotonin concentration. We see a response now in the upward current direction, but no response at the dopamine sensor. We can measure temperature at the same time. And because we have a third field effect transistor that is not functionalized with an aptamer, we can also detect small physiologically relevant changes in pH. And what you can see is when the pH changes, we also see a change in the response at the neurotransmitter specific Fett's. So by using this type of multiplexed architecture, we can then sort out which signals are due to pH and which signals are due to changes in the concentration of the neurotransmitters themselves. And finally, Chuan Zhen has been using these flexible designs to fabricate soft and flexible neuroprobes. Once again, you can see that he's able to fabricate those at the wafer scale. Again, here's a 2-FET design. You can see that they are conformal on soft material, and Chuan Zhen has developed a very clever way of implanting these soft materials into the brain tissue. So stay tuned. Those data are also coming out in publication form soon. So I just want to leave you with a final thought. We kind of started the talk today going all the way back to those early neuroscientists who were looking at brain cells in the early 1900s. But even as far back as the late 1800s, the great Charles Darwin, he authored this book on the expression of emotions in man and animals. And in this book, Darwin puts forth the hypothesis that animals and man, because animals and man come from a common evolutionary background, that the processes that are responsible for emotions in humans are likely very similar in animals. And if that is the case, and I believe that that's the case, then we can use animals as a way to investigate something that we tend to think of as being uniquely human. And I'll leave you just with this video. This is a test that we do in our lab. We have a lot of different behavior tests that we can use in animals to try to understand how they're feeling. In this particular test, we're asking the animal about its anxiety-related feelings. Mice don't particularly like to explore these open arms of this maze because they're dangerous, there are no walls, but yet they're compelled in some ways to explore their environment as they're looking for mates and food. And so the amount of time that they spend in these open arms versus these closed arms tell us how anxious a particular mouse is, or how anxious a particular group of mice are. And what we're currently doing now is we're integrating these neurochemical measurements with these types of behavior tests so that we can have real-time readout of what's happening in terms of brain neurochemical signaling as animals are making decisions about their environments. And so I'll stop there. Of course, we've had a lot of support from many different funding agencies to help this science forward. And I'd be happy to take some questions. Thank you.
Thank you so much. I'll bring this screen back up. Very good. Well, thank you for that lovely talk. We have a series of questions from the audience. Thank you to Chuan Zhen, working in the background with our center team.
We have poor Chuan Zhen always working, right?
Always working. Yes. How is the funding for brain science in the US and elsewhere?
Yes, that's a great question. As we were ending up on that issue of funding. With the Brain Initiative in the United States, we have seen increased funding specifically for the development of technology to try to think about these larger scale types of recording efforts. In general, funding for brain science has been pretty good. Both in the United States, there are large initiatives in Europe. Even in China, we see, I've certainly seen over my, the last 15 years visiting China, we see increases in support and effort in terms of investigating the brain. So I think, it's a good time to be in neuroscience. In the United States, miraculously enough, funding has been relatively stable over the last 10 or 12 years, even across administrations for brain science. And I hope that that's going to continue to stay the same.
Very good. The next question. Dear Professor Andrews, for the silicon Fett probe, what is its sensitivity? How do you calibrate it? And can it be used multiple times?
Those are great questions. The sensitivity of those probes are very similar to the sensitivity of the larger Fets. So the Fets that we were using for the characterization measurements, those are about 2 millimeters by 2 millimeters. The Fets on the neuroprobes are as small as 50 microns or 100 microns on a side. But it turns out that sensitivity doesn't scale linearly for field effect transistors, so making them smaller hasn't really impacted our sensitivity. So that still remains good. We calibrate those sensors. We typically calibrate them before we do measurements. In other types of measurement devices that we use, we would also calibrate the sensor after we make the measurements. And it turns out that those calibrations after the measurements are more important because at that point, the sensor has been exposed to tissue and the response is more like what we would expect to find in the brain. Can they be used multiple times? I mean, I think there's a possibility they could, but quite frankly, most of the sensors that we use are fabricated at large scales, and we would only probably use them one time. Even trying to implant them into the brain and taking them out would be tricky. So they're pretty much single-use devices.
And our third question, Professor Andrews, has there been any progress in seizure disorders with these advances in neurotechnologies?
Yes, that's a wonderful question. The advances that I'm aware of with seizure disorders have been mostly, in terms of the use of neurotechnology, have been mostly in the area of severe seizure. So this is for folks who have seizures that are serious and can't be well controlled by medication. And what we see for those patients actually is that they can have an ablation therapy where the locus or the focus of the seizure can be identified using a technology called neurocog to measure the electrical activity on the surface of the brain. And that's done before the patient receives surgery, and that enables the surgeon to produce a very, very highly localized lesion that hopefully doesn't hurt any of the other function, but stops the seizures.
Thank you. Our next question. I'm interested in your comments on the GRAB sensors that have been developed by Yulong Li's group. I think these are the genetically encoded GPCR.
Yes, I'm aware of them. Yep, GrabDA, Grab5HT. We're not working with Grab5HT, but we're working with S-Lite that was developed by Lin Tian. So this is a similar approach. As you heard about in Ed's talk, there kind of are two different general approaches to making measurements in the brain. One is to use optical measurements. There are some advantages in this case for these types of sensors. They can be genetically encoded to specific neurons, and their time resolution is fast. On the other hand, it's probably going to be difficult, at least as things are currently structured, to make highly multiplexed measurements of chemical neurotransmitters using these types of optical approaches. And one of the things that we can do with our implantable devices is to functionalize different Fets with aptamers for different neurotransmitters. We have learned, even when we look at just two neurotransmitters at the same time instead of one, that the hypotheses that we go into the experiment with are usually either incorrect or incomplete. And so I think this ability to multiplex is going to be really important. There's also this aspect where every type of sensing modality has its advantages and disadvantages. So you kind of have to pick the sensing modality that works best for the experiment that you're trying to do. There's not one that you would use to the exclusion maybe of the others. So I think there's some advantages of the optical sensors. I think there's some advantage of the implantable devices. That, you know, they may be slightly orthogonal, or you may learn slightly different things from those approaches. But I think they're both excellent approaches. And we ourselves are really excited about trying to use S-Light in some of our experiments.
That's a really great argument for how new tools open up new questions and help us, sort out what our, which of our hypotheses actually carry forward into reality and which ones we need to rethink.
Yep.
And then fifth question is, dear Professor Andrews, can you please summarize the key advances that scientists have made in terms of understanding the brain since the announcement of the Brain Initiative in 2013? There's no word limit to your answer here, I'll add.
Yes. Well, I mean, the short answer is, one of the things we learned is how hard it is to learn things about the brain. We actually kind of knew this going in. And so I don't think it's really possible to point to a single or two key advances, because we really haven't reached the goals that we set out for in the Brain Initiative, which were really to map large-scale numbers of neurons and to understand how information is encoded in both electrical and chemical signaling. I don't think we've reached that yet. However, what I'm really excited about, that I think has been enabled by the Brain Initiative and some of the discoveries that you've heard about today, is that we are seeing much more frequently now publications coming from neuroscientists where they are able to map circuits or parts of circuits that play key roles in behaviors. And so I think we're really starting to see that space open up. Whereas prior to 2013, or let's say, 10 years ago, we really weren't able to map even small parts of circuits or single transmitters or single groups of neurons onto specific behaviors. And we're really now starting to see that coming out of a lot of different groups. These tools, I think, have been really instrumental in enabling many neuroscientists to ask questions that they were not able to ask. So the building blocks are starting to emerge. What we don't yet have is really that holistic picture. For instance, if we take a complex function like, the feeling of anxiety, right? Let's say when we're faced with a difficult examination, we still don't really understand how, the brain, all of the different parts of the brain that are involved in that experience and that behavior are encoding that information. But we're starting to see parts of it. And the tools that we've heard about today, optogenetics, chemogenetics, neuroprobes, being able to map structure at high resolution. All of these things are helping neuroscientists to start to put these circuits together and to be able to ask much more complicated and much more pointed questions than we were really able to ask in the past.
Yeah, very good. Well, this would be the time if we're in Beijing, we'd walk across the stage together and I would hand you a plaque. But after this talk is over, I'll come downstairs for another coffee and bring you a certificate for the lovely iCANX lecture that you delivered today. We very much appreciate you and Ed and the insight you have for how to bring nanotechnologies and materials and new insight into the brain by developing new tools. So thank you so much for the talk this morning, afternoon, and evening, depending upon where our audience sits in the world.
Thank you.
Next week, we'll have a series of talks on liquid metals. Professor Alice Zhang will host. The following Friday, we'll return to our ACS Nano Rising Star lectures with Marquita Landry from Berkeley. We'll have work related to what we heard about today, as well as Martin Tuo from Iowa State and Jun Chen, our colleague here at UCLA. We look forward to welcoming you back and welcoming our new speakers to the iCANX family. Again, I want to thank Professor Alice Zhang for organizing and founding this wonderful series, and for Chuanzhen Zhao working in the background today to make all this a success. Thank you so much, and we'll see you next week.